Greetings, Life Sciences Grade 11. We are meeting again today uh, to just complete our last session of paper one. That is gonna be written now, end of term, end of year. For some of you who knows what they are here for, you are not lost. You are on the right track. You are on the right channel. Here, you are at home. Feel free, feel welcome. Just know that here is genetics guru. We are going to meet next year, January. Genetics guru, you will just be enjoying these sessions, all of them. I promise you, all these things are gonna be well explained. And today, without wasting any time, we want to quickly just go through this paper and just have a reflection of what is gonna be our exam that we are writing and as scope and exam tips. So please just note that you always need to read instructions. Instructions are very important when you are writing, and also check the time of the paper. It's 2.5 hours. So sometimes students they've got errors of writing without verifying how long is the exam that you are in. Also verify that instructions, read the whole instructions for the paper and the instructions per section and per question. So question one reads that it's a multiple choice question, which we are going to work on now. Uh, it is actually study the diagram below of an animal's skull and answer questions 1.1 uh, as we go. So now what is happening is that we are given this skull here, and then the skull is an animal that we can see that it has got it's an animal that we can agree and see that it has got the long teeth for tearing, which we call that it has got the large canines here, which identifies that the animal is actually it actually shows that it's an animal that has to do it has to do with tearing, it has to do with biting, it has to do with eating of meat. So now what is happening is the skull is saying the skull above is that of a dish. The skull above is that of a dish. You see, uh, it is actually, it can be an omnivore. An omnivore does not have this type of teeth. It cannot, omnivore because of large canines. The omnivore does not have large canines. It does not have, so it is not correct. Carnivore because of reduced premolar. We don't talk about the carnivore because of the premolar teeth. We talk about the what because we talk about the canines. I said there are large canines that are there. So an omnivore also does not have because of the canines and incisors. It actually is a carnivore because of large canines and sharp premolars that are there. It has got the canines that we say that it is actually for tearing and biting, which we can agree that these teeth identify that the animal is a carnivore, which has got large canines and the sharp molars. The function of the incisors of such an animal would be to dash. The function of the incisors of this such an animal, they are to actually bite and cut food, which is actually number C. They are there to actually bite and cut food. The incisors. I'm sure if you remember very well and check also from the videos which are on the channel, we have been looking at the skulls of various animals in terms of the ecosystem, the carnivore, the omnivore, and the herbivores. These animals have got the type of teeth that are determined by the type of food that they eat. So it actually has the incisors which are responsible for biting and cutting food. Uh, which one of the options listed below correctly summarize the waste products for each of the excretory organ? So we are saying we are given the excretory organs, excretory organs that are given here, and then each one of them has got its own specific uh, kind of what you call waste product that it produces. So if we look at this number 1.3, what is happening is that we should understand that the lungs are responsible. We are saying the lungs should be responsible for the excretion of carbon dioxide. It has to be the excretion of carbon dioxide, and the skin, it has to be what you call sweating, evaporative cooling, that we can also talk about in the near future when we talk about thermoregulation. And the liver is actually responsible for urea, and the kidney is actually facilitates urine excretion. So what we need to understand is that the lungs, they are responsible for carbon dioxide. The skin is actually produces or it releases water. We say this is for evaporative cooling, evaporative cooling of the body, which we need to understand that this all again it has to do with what? With the thermoregulation, temperature regulation, thermo thermoregulation. I'm sure that we are going to, some of you have seen that already, but we are going to talk about the human response to the environment soon. And then the liver, actually, it is responsible for urea, and then the kidney, which is urine. So our correct answer is what is B, which is carbon dioxide from the lungs, water from the skin, and then urea from the liver, and then urine from the kidney. So that is how it occurs, the waste that are the waste products from various excretory organs. It is recommended that athletes train at a higher altitude for at least 2 weeks before a major event. Altitude is the height above sea level, how high a place is, that is altitude, to increase the number of red blood cells in their body. The number of blood cells increases. The number of red blood cells increase to do what? I'm sure you remember the composition of the blood. What is it that we have in the blood? We talk about the blood platelets, we talk about the red blood cells, we talk about the white blood cells. So what is the function of the red blood cells? Red blood cells are specifically responsible for matters to do with oxygen. They have to do with we are saying the red blood cells, they actually transport oxygen around the body. So what happens is that the number of red blood cells increases to increase oxygen absorption because of the low oxygen levels in the atmosphere. So what we are saying is, as the higher you go, we are saying oxygen level has to be oxygen absorption because of low oxygen levels in the atmosphere. I'm sure you remember the carbon, the gases balance in the atmosphere, which we also explained in on metric hub when we talk about the composition of gases, or the composition of the atmosphere, how much uh, of each of the various gases are found in their proportionate percentages. So the answer is that is A, to increase oxygen absorption because of low oxygen levels in the atmosphere. That can be the right answer there. And then the reason for boiling the leaf in alcohol during a starch test is to do what? I'm sure, remember, we have got experiments that we have to do to determine if chlorophyll is responsible for, is necessary for photosynthesis. So what happens is that there are quite a lot of factors that we talk about that have to do with the life cycle or the survival of plants. We talk about oxygen, we talk about carbon dioxide, we talk about chlorophyll, we talk about sunlight and all those. So now, when we boil the leaf in alcohol during starch test, it is actually done in order to remove the chlorophyll in the leaf. So that chlorophyll, the green pigmentation, chlorophyll to remove that is green pigmentation in a leaf. Green pigmentation in a leaf is removed by what? By chlorophyll, meaning by alcohol. Boiling the leaf in the alcohol, it actually helps to remove the pigmentation, which is chlorophyll. So therefore, we can be able to see the idea of starch test, the iodine solutions and all those. So We are saying it is done in order to remove what? To remove chlorophyll. And then if the blood pH decreases, the kidney will dash. The blood pH decreases. We are talking about the potential hydrogen. We said last time that the pH, if the pH decreases, it also causes the acidity of the body. I mean, it increases the acidity levels or the acidity conditions of the body. So now, if the blood pH decreases, it actually increases the absorption of urea in the body. It's not doesn't. We are saying it does not increase the absorption of urea, decrease the secretion of hydrogen ions into the tubules. It actually increases the reabsorption by bicarbonate ions in the blood. What happens is that when we look at pH, when pH decreases in the kidneys, actually the reabsorption of bicarbonate ions is gonna increase. That is what happens when pH decreases. And then study the list below and answer the questions. Water, carbon dioxide, hydrogen, ions, and radiant energy. Which of the above listed reactants are required for photosynthesis? When we look at photosynthesis, we need to understand that water must be present, carbon dioxide must be present, and also energy, which is for what you call uh, the solar light or a phototropism or a photo as in terms of the light availability. So we need to have water. Water must be present in order for photosynthesis. Obviously, the survival of plants. Carbon dioxide, which is the exchange of oxygen and carbon dioxide by plants and living organisms. And then we are saying the what? The light, the radiant energy, which we say light. So all of this, it should be therefore mean that the correct answer is what? Is A. Water is actually needed. Active uptake of water by the roots, carbon dioxide in order to facilitate gaseous exchange. I'm sure you understand environmental science when we look at the essence of plants and vegetation in terms of the balancing of the removal of greenhouse gases in the atmosphere. So it is very important that you understand that carbon dioxide is an essential gas that is needed in terms of survival, in terms of the manufacturing of food by plants. I remember as well as you can look at it, the summation of photosynthesis, the equation, as you can look at it, I'm sure that you can see carbon dioxide, it forms one of the key elements that is very important in there. So a plant stomata grows when temperature exceeds 30 degrees. We are talking about the small holes on the leaf, small holes on a leaf is what we call the plant, is what we call the small holes on a leaf is what we call stomata. And then what is they close when temperature exceeds 30 degrees. So therefore it actually try to reduce, this has to do with trying to reduce, it actually helps to reduce transpiration in order to avoid too much loss of water. So what is the most likely factor to limit the photosynthetic rate after this happens? When a plant stomata closes when the temperature exceeds 30 degrees Celsius. So what happens is that what is the factor that the most likely factor to limit the photosynthesis rate? It is we can't talk about light intensity. Light intensity, it does not has nothing to do with the stomatas. It has nothing to do with the stomata and also oven oxygen concentration. Remember that this is a byproduct, so we don't need it. It is produced after photosynthesis. This is a byproduct of photosynthesis, so we cannot worry about that. Temperature has nothing to do with with the photosynthesis, because what we need to look at is that like from the factors that we have discovered above, the carbon dioxide concentration, because it is actually exchanged through the stomatas, the opening and closing of the stomatas on the leaf, which is actually gonna be the reason that it can actually affect the rate of photosynthesis. And then the diagram below identifies the process occurring at X and Y between both tissue cells and capillary. We are looking at carbon dioxide oxygen concentration and exchange, gaseous exchange, which is shown here. So uh, the diagram identified the process occurring at X and Y between both the tissue cells and a capillary. So we need to find which one is that. So number this is number 9. So what is happening is that it actually shows gaseous exchange in the passive process. It actually shows gaseous exchange. That is to say, oxygen and carbon dioxide exchange that can also be illustrated by the process which is gaseous exchange and passive process, which is being shown. Again, if you check on metric hub, this has been, we have the same diagram where the explanation is given that we talk about the passive process and gaseous exchange, which takes place, which is occurring as it is indicated in the diagram above there. So we need to give the biological terms for the following descriptions. Uh, the organelle in plant cells that contains chlorophyll. Chlorophyll in a plant cell, it is the organelle in a plant cell which contains chlorophyll. It is actually the chloroplast. Chloroplast. The chloroplast, it actually contains chlorophyll. And then breathing muscle responsible for raising the rib cage during inhalation. It is the intercostal muscles. I'm sure that if you can remember this process, we are saying muscle responsible for raising the rib cage, which is actually to say that the responsiveness of the cavity with regards to gaseous exchange in particular to inhalation. We are saying it is actually controlled by the intercostal muscle. Inter intercostal intercostal muscle. And then the structure, tube responsible for transporting urine from the kidney to the bladder. It is actually kidney, the tube that carries urine from the kidney to the bladder. It is the ureter. It is the ureter that is responsible for that. This is it is the ureter, a structure in the lung where gaseous exchange occurs. It actually occurs in the alveoli. I'm sure this also, the diagram was well explained in during the course of the term, to say that the alveoli is actually responsible for gaseous exchange in the lungs. Those micro small holes that are found, which allows gaseous exchange in the lungs. As we go into that, the main photosynthetic tissue of the leaf, it is actually the main photosynthetic tissue of the leaf. It is actually the palisade, which we can say the mesophyll. It is the palisade layer, or the mesophyll. And then the breakdown of a water molecule into hydrogen and oxygen atoms during photosynthesis. Water molecule it is actually the photolysis. Photolysis is the breakdown of water molecule into hydrogen and water into hydrogen and oxygen atoms during photosynthesis. It is photolysis. And then the protective double membrane enclosing the lungs. The protective double membrane enclosing the lungs, it calls the pleura. It is the pleura that is actually responsible for protecting the lungs. It is actually a double membrane enclosing the lungs. And then gas required for aerobic respiration. We are talking about oxygen. Oxygen is the gas that is needed when we talk about aerobic respiration. Oxygen. Patches of a cell in the pancreas that secretes insulin and glucagon. It's called the islet of Langerhans. I'm sure this one also, we are going to continuously talk about this as we go to grade 12 when we talk about the balancing of the sugar diabetes conditions which are caused because of excess insulin. I mean excess sugar levels that or glucose that is unconverted into its storage form. So the specialized cells that secretes these hormones is called the islets of Langerhans. Islets of Langerhans. They actually secrete the insulin and glucagon. Insulin, it converts excess glucose into glycogen, and the glucagon actually responsible for converting that glycogen back into what? Into glucose when the body needs more energy. That's what you need to understand. So I have got also a nice diagram that I have explained last time, which I showed the irreversible reaction between insulin and glucagon. Insulin decreasing the glucose levels and glucagon increasing it. We are supposed to see that it applies to B or none of the above. Structure or structures which prevent food particles from entering the lungs. The food particles are actually avoided from entering the lungs by the epiglottis, which is B only. It is actually B only region of the alimental canal where most water is reabsorbed. Water is reabsorbed. At the reabsorption of water, it takes place in the what? In the alimental canal. We talk about the small intestine. It is the small intestine which is B only. Lined with ciliated epithelium. It is lined with ciliated epithelium. What is that which is lined with ciliated epithelium? It is actually, we say that there is it is also the bronchi and the bronchia. The bronchi and the trachea, they are both lined. So it is both A and B. Both A and B. And then hormones responsible for osmoregulation. We talk about balancing of water. The balancing of water, it can also be it can be balanced by B only, which is antidiuretic hormone. Aldosterone hormone is actually responsible for salt balancing, but we are saying it is actually the best answer is antidiuretic hormone, which is responsible for balancing of water in the body. The antidiuretic hormone, it is responsible for balancing of water in the body. We have got question 1.4, which identifies models of the lungs below used to demonstrate mechanism of breathing. We are seeing the mechanisms of breathing here, which is given structures here, A, B, C, D, and what structure in the respiratory system is labeled is label A, representing what trachea in the respiratory system is label A representing. As you can see, A is representing the trachea, which is the pathway, the passage. It is representing the trachea. A represents the trachea, and then describe the role played by D during inhalation. D, the role played, we are talking about the intercostal muscles. It is actually, we are saying which is actually showing the diaphragm. It actually identifies, it identifies the diaphragm. D, we are saying that it is the diaphragm. It is the diaphragm. It contracts, increasing the volume of the thoracic cavity. It actually, the diaphragm contracts, it contracts, decrease, in order to increase the volume of the thoracic cavity, actually decreasing the pressure in the thoracic cavity. The diaphragm contracts in order to decrease, the pressure. It actually to decrease the pressure in the thoracic cavity. Pressure in the thoracic cavity. So actually, I'm sure that if you can remember last time again, we talked about this. We talked about this question. The diaphragm actually, it has to cause the increase or decrease of volume in the during the gaseous exchange. So when the person breathed in, when you breathe in, it goes up, which actually reduces. When you breathe in, sorry, it actually goes, when you breathe in, it is actually increasing the volume there, and then also to decrease the pressure in the thoracic cavity. And one thing that you need to know is that the volume increases. Volume increases in thoracic cavity. Obviously, you can demonstrate this by yourself. Try to breathe in and identify the changes that will take place there. And then 4.3, give 2 reasons why the model does not truly represent the human gaseous exchange. This one, it cannot fully show human gaseous exchange, actually, because when you look at it, it's just a demonstration. So we can need to agree that the size of the model is immovable. Size of the model are immovable, unlike unlike if it is a human chest, it moves up and down, unlike a human chest that can move up and down, that can move up and down. And then we need to understand again that which can actually, when it moves up and down, it actually shows the contraction and relaxation of the intercostal muscles if it is a human chest. And then the spaces between the lungs and the walls of the thoracic cavity is very small in the model. If you can look at that, the space between the lungs and the space between the lungs and the walls of the thorax is very small. As you can look from the model there, which shows a large space between the balloons and the glass, which is something that is different. So we are saying two space between the lungs, the space between the lungs and walls of the thorax is very small. It's actually small. If you can say it's actually small, uh, uh, actually the model shows a large space between the balloons and the balloons and the glass jar, whereas the model, it shows a large space between the balloons that we are seeing there, the balloons, between the balloons and the glass jar, balloons and the glass jar that is given there to illustrate the human cavity. So it's one other big difference that we can see there. In human body, the diaphragm is actually dome-shaped and flattens during inhalation. In human body, in human um, in the human body, the diaphragm the diaphragm, actually what we can see is that diaphragm, the diaphragm is dome-shaped. It is dome-shaped. It is actually dome-shaped and flattens during inhalation. And it flattens during inhalation. It flattens during inhalation, which again in the model the rubber sheet is flat and is pulled down to represent inhalation. And the model, the rubber sheet is flat. The rubber sheet is flat and is pulled down to represent inhalation. We can also talk about things to do with in terms of that in the body there is limited movement of lungs or pressure exerted on lungs. In the model there is a lot of space available. I mean in terms of pressure in the body there is limited movement of lungs, which actually you can see that actually there is pressure exerted on lungs in the model is a lot in order to cover up the available space. So greater pressure is placed on the balloons in terms of the model that we can see the amount of pressure that is placed in the balloons there is quite a lot in order to cover up the space that is in between the glass jar and the balloons. But as you look in a human body, that cannot be the same. And then also balloons are actually showing that there is one large circle and lungs is not hollow shaped as the way it is shown there. The lungs are not hollow shaped. That is one other key area of concern that we can give. The diagram represents part of a human gaseous exchange system, which is exactly like the way you see here. We are looking at the original versus what has been given there as the model. So what we need to look at here now is that what we need to look at here, we need to look at uh, various questions that would arise here. Let's just see what are they saying. Supply suitable labels for the following structures: B, B, C. What is B and C? B and C. If we look at this one, B, we have labeled the previous one. We said that as you look from the trachea, we remember A is the same thing that we labeled as the trachea there. So B and C, B actually shows the bronchus, which are actually showing the cartridges, or the it actually shows the bronchus here. B shows the bronchus, and then C it actually shows the rib. We can see that C is the ribs. C is showing the ribs that is showing the rib that is being shown by C there. So B is the bronchus and C is the rib. And then the next question is saying, so it is bronchus, and then it is the rib that we can see from the diagram there. Explain two visible reasons why the above structures could be considered an effective gaseous exchange surface. Two visible reasons why the above structures can be considered an effective gaseous exchange surface. So we are saying, one, if we look on these structures that are shown here, why can they be considered effective gaseous exchange surface? One, we can talk about that the presence of a double membrane which provide protection against double membrane that you are seeing here, the double membrane along the lungs. We talked about that in the previous questions, that there is the plural, which is actually a double membrane that protects the what? Protect against friction. We are saying presence of the double membrane to protect the lungs against friction. Against friction, it actually shows that it is a good surface area for gaseous exchange, the plural, which we are saying that it helps in that. We have got also uh, uh, the trachea and the bronchi breathing muscles and intercostal. They allows a movement of what? Of air in and out. As you can see here, we are saying the trachea. We have got the trachea there. We also have the bronchi, which we also see here. And then we can talk about the breathing muscles, and the diaphragm allows movement of air, and there is effective ventilation. Effective ventilation that allows free movement of air in and out of the lungs, in and also outside the lungs, that we can actually see. That can also, we can see, we also have there is also uh the large surface areas that are also there, that you can see the large surface area which can also be seen there. Large surface area, which we can also have main alveolus are present to increase the surface area for gaseous exchange. That you can see the alveolus are there, the alveolus are there, and main for effective gaseous exchange. Effective gaseous exchange can take place. Or we can say that this diagram, it is actually considered an effective gaseous exchange surface because of the factors that we are listing here. It is an effective gaseous exchange surface, effective gaseous exchange surface due to these points. So the diagram that we are seeing is an effective gaseous exchange surface because of this reason that we can give here. These are the reasons that can make it an effective gaseous exchange surface. And then give a letter and name of the structure that contains the cartilage, the cartilaginous cords, which vibrate for sound production. Number 1.53. The letter, check here. I always want you to take note to say we have got 1, the letter, and also 2, the name. So we have got 2 questions here. So give the letter and the name of the cartilaginous cords which vibrate for sound production. So sound production, it is the larynx that actually represent that. So we need to identify from the diagram which one is the what is the larynx. So we can see that this is G. G is the right answer. G, which we say that it is the what? It is the larynx. It actually helps in terms of, it actually helps in it actually vibration for sound production, and then contains C-shaped cartilaginous rings. The C-shaped cartilaginous rings, we also need to understand that these are found on the trachea. So we need to see that these are found on the trachea. So which letter represents the trachea? Which letter represents the trachea is what we need to find out. So the trachea is represented by A. We have got the trachea here, which represents A. So it is A. The trachea, it is the one that identifies what is given there. And then we want to quickly go to question B. Section B, scientists conducted a study to determine under which color of light elodia, which is waterweed, grows best. The experiment was conducted as follows. Apparatus was set up in the diagram below. 3 experiments were set up, each under different color light: blue, green, and red. The distance from light source in each remained 30 centimeters. So we want to see the distance from light source remained 30 centimeters. So we are given here that oxygen bubbles are Elodia plant. So the average time taken to produce 20 oxygen bubbles was recorded in the table below. So to produce 20 oxygen bubbles, so time in seconds to release the 20 oxygen bubbles. In blue, we have got 40 seconds, 160 and 80 seconds are given here. So the question is saying draw a histogram to represent the data that is in the table above. So when we are drawing a histogram, the question is data presentation class. When you are given this question, you should always know that there is a title. There is always a title. So what is the title of this data of this histogram? We need to look at the amount of oxygen, the amount of time in seconds to release 20 bubbles of gas in different colors. You can say time taken to produce 20 bubbles of oxygen, 20 bubbles of oxygen in different colors of light, in different colors of light. So that is the title. We say that the title must be able to capture the dependent and independent variables. They must be able to be seen. So we are measuring the 20 bubbles of oxygen produced in different colors of light. So that is our title. So now when we are drawing, one thing that I want you to look at again is that this is a histogram. So a histogram meaning to say that the bars must be touching each other, and at the same time, we need also to consider the highest, the lowest and the highest limit. So we have got this as our lowest, this is our lowest variable, and then this is the highest. So what does this do? It helps us to set a scale which can be used to draw a graph that can be standard in our question here. So our lowest value is 40, and our highest is what? Is 160. So we can be able to choose a suitable scale that can be able to represent this set of data. So I can say this can be drawn to say that here we have got time in seconds. We have got time in seconds on the vertical axis. We have got time in seconds. Time in seconds on the vertical axis to release 20 bubbles to produce 20 bubbles is on our vertical axis. And then in this other horizontal axis, we also need to know that these are the colors of light. We have the colors of light on the horizontal axis. Colors of light on the horizontal axis, so we can be able to draw our graph. So I was saying, for me, I can take an interval of if I take an interval of 40, I can say 40. If I take an interval of 40, and then I say 80, 120, 120, then I can have 120 plus 40, 160, is to say that is our scale. 40, we have 40, 80, 120, 160 is my interval there. So we said that from the set of data, we have got the blue light, it took 40 seconds. Blue light, it took 40 seconds. So I can be able to say the blue light, it took 40 seconds. So therefore, this can be 40. I always want you to understand one thing, that you should be able to give the readings on top of the graphs. You must always be able to read, to say that each bar must be marked to say what does it represent. So we have got 40, and then this is blue. So we have got blue light here. And then we have got the next one, which is 80. Remember, these bars are going to touch each other because it is a histogram. and the width must be also be the same. So we have 80 there. The 80 it is representing, we have 80 that represents, 80 represents, sorry, I can take the second one is green, which is 120, 160, sorry, it's 160. We have to say it's 160, not 80, 160. So 160, we can go here. So this is 160, and then it is green. And then finally we have got 80. 80, which is red. So if you can do this, when you can do it this way, your marks cannot be doubted. What I said is that you always need to be able to label the axis. So the axes are labeled is colors of light on the horizontal and time in seconds on the vertical. And then there is a suitable scale there of 40. We have got our scale of 40, which is actually given 40, 80, and then 120, 160, something like that. Our graph can be just so readable and clean like that. So what I'm saying is the title is also given here. We have our title given. So what is very important is for you to be able to know that all of these sets of data or all the given information must be suiting towards a histogram. So this is a histogram. Hence, we can say that these are the readings that we can see: blue, green, and red. 120, 80, 140, 120, and 80. These are the markings that we are given there. And then why did the scientists use oxygen bubbles production to look at the growth of the plant? I'm sure that you know that when we look at this one, oxygen bubbles to look at the growth, to measure the growth of plants, it is actually because oxygen bubbles production indicate photosynthesis that is occurring. And then photosynthesis is the one that is responsible for the growth of what of plants. Oxygen production confirms photosynthesis. It confirms photosynthesis. And then photosynthesis is the one, photosynthesis, which is the responsible process for plant growth. When you talk about plant growth, it is measured because of, if a plant is alive, it must be able to make its own food. So oxygen is a byproduct of photosynthesis. I think that one we are quite sure about that. So for us to see that oxygen is being produced, it confirms the livelihood or the survival of the plant. So we are saying that that is one key thing that you should know. And then write the ratio for number of bubbles under green light compared to red light. So we can just say green as to red, and then we put the numbers of green as to red, green light as to red light. So green light is given 120, green light is given 160. So this is 160 as to red. Red is what is 80, red 120 as to what is to 80. So we can actually remember that ratios can further be simplified into their simplest form. You scientists, I'm sure that you know, we can now do that. What is needed here is for you to be able to simplify this by what? By dividing by what? Dividing by that 80. We divide by 80 there. So we divide 80 into 120 into 160, it is 2. And then we divide 80 into 80, it is 1. So 2:1. This is the simplified. It is just a simplified ratio of 160:8. We simplify it by dividing by 80. It is simplified by dividing by 80 class. That is what is yours. That is one thing that you should take note of. It is simplified by dividing, or we find the simplest form of any given number by seeing likely chances of further division. So therefore, if there is any chance of dividing, we further divide the answer in order for us to get this, the final correct one. So the answer is 2:1. It is 2:1. That is the final answer that you should note. Which light source should be recommended to grow? Which light source should be recommended to grow plants under? Which light source should be recommended to grow? Which light source should be recommended to grow plants under? Which plant, when we look at these, which light can we recommend to grow plants under? So as we can look at this one, we can say the light which we can recommend to grow plants under, it is actually, I can give to say that the one which has got the highest amount of oxygen, as we can say that oxygen signifies life. So for me, I can actually agree that the one which has got the highest amount of oxygen, it is what? It is green light. I can say green light due to high growth rate indicated by amount of oxygen indicated by the amount of oxygen produced. It has the highest light. It has the highest oxygen content. We have got the highest oxygen is produced on the green light. So that is one key thing that we must consider in these kind of quizzical questions. A serious motor vehicle accident resulted in a child having a perforated diaphragm, which is diaphragmatic hernia. His abdominal organs were pushed into the thoracic cavity. Symptoms included difficulty in breathing and a rapid heart rate. So we have got the abdominal cavity or abdominal organs in the diaphragmatic hernia that is shown here. So the question is saying, discuss how the hole in the diaphragm caused the diaphragmatic hernia will affect the inhalation. You see, these questions are just more or less the same. So one, what is happening here is that the space in the thoracic cavity would be limited because we are saying that it is pushed inside. So it reduces the thoracic cavity volume. The thoracic cavity or volume is reduced. It's actually reduced. That is one. The volume is reduced. So it means less oxygen or carbon dioxide, and less oxygen can be taken in, and then due to abdominal organs pushing into the thoracic cavity, It's actually limiting inhalation and the ability of lungs to fully expand, because the abdominal organs are pushed inside. The abdominal organs are pushed inside. They are pushed inside and they limit inhalation. Therefore, they limit inhalation. And the expansion of lungs. Expansion of lungs to their fullest size. Lungs cannot be able to expand to their fullest size. So describe why the symptoms of a rapid heart rate would occur. Why the symptoms of rapid heart rate would occur. So this can be due to difficulty in breathing, damaged diaphragm, may cause difficulty in breathing. It may cause difficulty in breathing. The damaged diaphragm, remember we said that the diaphragm is damaged, therefore it can cause difficulty in breathing. It can cause difficulty in breathing. So carbon dioxide in the blood increased, which was detected by what? By the chemoreceptors of the aorta. Carbon dioxide in blood increased. Carbon dioxide in the blood it increased. So therefore what happens is that it will not be what? It will not be exhaled, because it is not effectively exhaled. Is not effectively exhaled. So this can be detected by the what? By the receptors of the aorta. Such can be detected by the receptors. I'm sure that you remember that these are the chemoreceptors of the aorta. Which stimulates the medulla oblongata, stimulating medulla oblongata to send impulses to the muscles, to send communication impulses to the heart muscles. Impulses are sent to the heart muscles so that the concept so that it can also to cause increase in what in contraction of the heart muscles. Increase in contraction increases in contraction of heart muscles. So these are the damaging of the diaphragm actually is causing the breathing problems. So it is affecting the gaseous exchange, which would always cause the stimulation of medulla oblongata to communicate impulses to the heart muscles, which increases the contraction of the heart muscles that would actually cause this rapid heart rate in order to balance the gaseous exchange that we are talking about. Researchers wanted to determine if living organisms require oxygen for aerobic respiration. Oxygen uptake can be measured using a respirator in the setup below. The apparatus consists of two flasks, one containing the living organism and other glass beads. Both tubes contain the same volume of sodium hydroxide. And then once the apparatus has been set up, the movement of the colored liquid in the manometer actually will indicate how much oxygen is used. The decrease in oxygen in the tube increases the pressure, causing the colored liquid to move up the tube. So the distance between the liquid in the manometer over time was measured to indicate the volume of oxygen taken in by the insect per minute. So findings were recorded in this table as we can see below here. So we are given movement of the dye in the manometer over time. Flask A, minutes, 15 minutes, 30 minutes, 45 minutes, and 60 minutes. So we are given the syringe containing air, and then the closed tape here, the insect, the metal gauze. We also see the manometer containing the dyed fluid with the sodium hydroxide, which is also shown here. And then that is sodium hydroxide solution, and the glass beads, same mass as insect, and filter paper, which is also shown below the rubber stopper and containing syringe air. So the question is saying we are asked to identify we are asked to see what is the aim of this investigation. As you can see, we are saying there is living organism here, which is the insect, and we also have the glass beads, that are also there. So now we need to determine what is the idea of this investigation. So as you can see, this is actually, if you can read on the narrative, it shows that the distance moved by the liquid over time measured to indicate the volume of oxygen. We will need to measure the volume of oxygen taken in by the insect per second, per minute, finding shown in this table. So this is to determine if living organisms require oxygen for what? For aerobic respiration. To determine if living organisms requires oxygen, if they require oxygen for aerobic respiration. the aerobic respiration. The setup of the apparatus says it, or it actually gives all the answers that we are seeing here. Determine if living organisms, they actually require oxygen for aerobic respiration. So identify the dependent variable. Identify the dependent variable, the dependent and independent variable. So our investigation is actually on the amount of oxygen versus the absence or presence of living organisms. So oxygen is actually dependent on the what? On the living organisms or non-living organisms. So the dependent, it is actually aerobic respiration. Aerobic respiration, it actually shows the dependent, and then we are saying the presence or absence of living organism is the independent. The presence or absence of living organisms are actually an independent variable. So the next question is saying calculate the percentage increase of oxygen consumption from 30 minutes to 60 minutes from flask A. The oxygen percentage increase of oxygen from 30 to 60 minutes in flask A. In flask A, from 30 to 60 minutes, 30 we have got from 30 we have got a 30 flask A, we have 13, we have got 13, and then at 20 minutes we have got 28. So percentage increase, it is always going to say that final minus initial over what? Over initial times 100. Percentage increase is equals to final minus initial over initial multiplied by what? Multiplied by 100. So our final, as we can see, we found out that the final is 28 minus initial 13. So it is 28 minus 13 over 13 multiplied by 100 over 1. And then that can give us to say 13 minus that one over that. We can give it through to say that this is equals to 13 minus 5, 3, 8 minus 3 is 5, and then is 15 over 13 multiplied by 100, which can give us, 115.4%. Percent 115.4% is the percentage increase, because we are saying final minus initial over initial times 100. And then we are given, explain one way how validity was ensured in the investigation. The validity, we are saying the validity was ensured by both insect and the glass beads have the same mass, and therefore starting position you have the same pressure. We have got one, both the insect and the glass beads They have the same mass. They have the same mass, which actually shows that same pressure they also indicate that they have the same pressure at first. And same pressure from the start. They have the same pressure from the start. You can also say that we have got the control, which is also present. The control of the investigation is all the same conditions. The control of the investigation have the same conditions. It has the same conditions. It has the same conditions, except for the independent variable, except for the living organism. So the living organism is obviously, we remember that during an investigation, we must not cause the organisms to die. So it must have the capacity to be able to continue to survive. Respiration occurs in both aerobic and anaerobic conditions. These processes differ in plant and animal cells. In plant cells, aerobic respiration has been shown to hold several key economic uses and benefits. Describe the process of the Krebs cycle. The process of the Krebs cycle. What happens with the Krebs cycle? I'm saying this is one key area that is we have been discussing and identifying ever since, again, during the course of during the course of classes. So one, we must understand that pyruvic acid is broken down, releasing hydrogen atoms and carbon dioxide in the presence of oxygen. We should understand that pyruvic acid is broken down. It is broken down. Pyruvic acid is broken down to release hydrogen atoms. To release hydrogen atoms and carbon dioxide. And carbon dioxide in the presence of oxygen. In the presence of oxygen, and then small amount of energy is released, resulting in small amount of energy, resulting in a release of small amount of energy. That's the Krebs cycle. It actually identifies the breaking down of pyruvic acid to release hydrogen atoms, carbon dioxide in the presence of oxygen. That is what you need to understand in this respect. So we need to look at this is carbon dioxide. We need to look at how does aerobic respiration differ in plant cells compared to animal cells. Plant cells compared to animal cells. How does aerobic respiration differ in plant cells compared to animal cells? So what we need to understand is that in animal cells, pyruvic acid is partially broken down into lactic acid. In animal cells, pyruvic acid is incompletely broken down, which is it is partially broken down into lactic acid. It is partially broken down into lactic acid because of the fermentation process. Due to fermentation of the lactic acid, due to fermentation, and then in plant cells, pyruvic acid is partially broken into ethanol and carbon dioxide. In plant cells, pyruvic acid is partially broken into ethanol. It is partially broken into ethanol and carbon dioxide. Which actually due to the alcoholic fermentation. Alcoholic fermentation actually causes in plant cells to partially break pyruvic acid into ethanol and what and carbon dioxide. So now what you need to understand is that alcoholic fermentation can be used in alcohol, which can be sold for income. It can also be used to make dough to rise, and then that are some of the what alcoholic fermentation. Alcoholic fermentation can actually be used in to form alcohol, which can be sold for what for people's living, which is making money, and then it can also be used to make bread dough rise in order to increase a greater volume of product which is produced at a greater profit also. Discuss two economic benefits, which are the economic benefits here that I'm already mentioning to you, that the economic benefits of use of aerobic respiration in yeast. Like I said, that alcoholic fermentation can be used to form alcohol, which can be sold for income. Alcoholic fermentation can be used to form alcohol, which can be sold to make income. That can be sold to make income. And then we can also say that it can also be used for raising of dough. To raise dough. in bread production. When bread is being made, we can also use this yeast in raising of dough in order to make uh, bigger bread production to increase its volume. And therefore profit. Therefore profit can also be increased. And then list one symptom of prolonged aerobic respiration in muscles. One symptom of prolonged respiration, one symptom of prolonged aerobic respiration in muscles. This mostly results in what? In fatigue or spasms. We can talk about fatigue. We can talk about fatigue or spasms. Fatigue is a symptom of aerobic respiration. So gaseous greenhouse gases or greenhouses are enclosed structures used to grow plants under controlled conditions. Carbon dioxide levels are kept high during the daylight hours, so optimal photosynthesis can take place. The graph below displays the carbon dioxide concentration over 24 hours period, 12 o'clock until 4 o'clock. Carbon dioxide was pumped into the greenhouse. It was pumped into the greenhouse. So the carbon dioxide concentration in which is given here, and then carbon dioxide concentration, and then there is time which is 24 hours, that is shown from 0 up to 24. So the question is saying, the question says, one, name two other environmental conditions that can be controlled in a greenhouse. In a greenhouse, obviously light intensity is controlled again. Light intensity, the strength of light that enters is controlled. Light intensity, it is controlled. We can also control temperature. Because there is a shade, that shade is controlling temperature. We can as well, we can also control water or rainfall.